I'm not sure yet. This associate is hiding. Actually, there's two. There's another one down below. Looks like a shrimp to me. You can see the one on the bottom is a shrimp. Yeah, we have two shrimps here. I believe several Chrysogorgia species often have shrimp associated with them. But we also see squat lobsters at times as well.
Sometimes it's the best you can do with these kind of ephemeral things.
Okay, and when you're done filming here, if we can pan right.
Andrew, were we at full zoom on that?
Oh, wait, just a second, guys. Go ahead, Tim.
Were we at full zoom on that lower shrimp before?
Yes, we were. Full zoom. Do you want to creep up and get a closer look?
I don't know how difficult that is, but
can we?
It would be nice from my perspective.
Is there a way that we can creep up a little bit to get a closer look at that shrimp? We can give it a try. There's another different coral in the background than we've seen on the upper right. Might be another Chrysogorgia.
There may be another
Chrysogorgia
up on the hill there. I can't tell.
And we have, I think most of the stocks in the background are small Paramaricia colonies. As we move up and zoom in, you'll be able to see those as well. So several different octocoral species here. No, Brendan's got it. Thanks. Is that a shrimp in the foreground of the Chrysogorgia as well?
That video. Try to get closer if you just looking back.
And we are at a depth of 1768 meters. Our local time is 10:53. Today, the 16th of July, we are surveying the east wall of Atlantis Canyon off the coast of the northeastern U.S. We're currently off the coast of, on the continental slope from New Jersey up through Rhode Island. In a very large region here, we are surveying canyons, submarine canyons off the coast. Trying to understand the geomorphology and the geology, as well as the benthic communities associated with these canyons. We've been the past few days surveying deep sea coral communities and sponge communities, noting a high diversity of corals, deep depths in these canyons along the walls. We've also visited a few cold seep sites and confirmed chemosynthetic communities off the coast here, some of the most northern distributions on the Atlantic margin on this side, on the western Atlantic margin.
That is correct, and we've also done a couple of dives focused on run by USGS, United States Geological
So
Survey,
pan down.
focused on natural hazards on the seafloor. So looking for large movements or slumps or blocks that have the potential to move or have moved.
Hey, watch, so he's
If you can pan down, there's a good view of one. Oh,
this one?
Nope.
This is
oh, is there another one in there? Yep. And we're just trying to, right now, get good close imagery. There's a few associates, crustaceans in this Chrysogorgia coral. It's difficult to see. Oh, here it comes. There's one out now. This was one of the same individuals that was in the coral, embedded in the coral. What do you guys think there, Tim?
Yeah, we have 2 amphipods living in among the branches, and also a shrimp out here, as we often see, perhaps swimming in the lee of this coral. If there is the lee.
these skids just shifted on the bottom,
So in the foreground here, we have a mysid. And within the Chrysogorgia, there is 2 amphipods. Go ahead, Tim.
No, I was just saying, if you've been following along, you know that there's, we often see amphipods, small white amphipods walking amongst the branches and the pods on these corals. But these amphipods seem to be different than what we've seen before. They're also much larger than what we've seen before.
On this shrimp species, I don't believe we've seen this yet on this cruise.
Yep, I would agree with you. That's true.
we're on some global, so the squid flips on axis.
All right, we're getting really good close imagery here of a shrimp. I'm good with that. We can come partial out. Okay.
Pick up the,
Yeah, and then if you can pan up and get a close-up of all those 3 the corals. There look like sticks coming out. Yep. Looks like we might have a different species in the back here too. Maybe another black coral. So we'll do these 3 and then. Again, I'm noting that there are several small colonies here. Is that red one maybe a small bubblegum or a Swiftia? It's hard to see.
I think we might be a little bit deep for the bubblegum, so it's more likely a Swiftia. And I think that may have been a Bathypathes black coral in the background.
So we're just getting resituated here?
You want those 3 yellow ones again?
Yeah, yep.
Okay. I had them, but then slipped.
Yeah, so we just slipped off this scarp. We're going to go back in and get a close image of the 3 very small coral colonies. And likely are, relatively recently settled onto the seafloor. although these corals that we're viewing here can live potentially hundreds of years old. We're still learning about the ages and the growth rates of corals in the deep sea.
I don't know what the ages and growth rates on Bathypathes might be. If they're anything like the other deep water species, then they would be extremely slow growing.
How about Chrysogorgia? Has anybody done any age work on Chrysogorgia?
Not that I can recollect off the top of my head.
I don't believe so, Andrea.
They have pretty small bases, and they
Yeah.
have very thin central branches.
And that's the same thing with the Bathypathes. It's typically, whenever I've seen them, they're quite small. So it'd be hard to get good radiocarbon age estimates off of a small stock like that.
And so you have to take cross sections, slice the stalks fairly thin,
Yes.
and then you, what type of technologies do you use to age it? Do you use laser ablation?
No, there are a number of different ways that you can go about it. Typically what we found with the deep sea corals is that using radiocarbon is one of the better ways. There are some caveats to that, in that over the last couple of hundred years, the radiocarbon signature in the ocean and in the atmosphere has not changed that much. And so if you have very young specimens, or specimens are only a couple hundred years old, they're hard to date using radiocarbon. The other interesting thing from a radiocarbon standpoint, when we're talking about these deep sea corals, and particularly the ones with a proteinaceous skeleton like the black corals or the gorgonin portion of the bamboo corals, is that they're actually incorporating young fresh carbon that is directly exported out of the surface waters. So the carbon signature in the skeleton of those corals is in fact a signature of what's happening in the surface ocean. So it only takes, a couple of days to a couple of weeks for that particulate organic matter, or that marine snow that we were talking about yesterday, to sink from the surface to the deep ocean. And that's the primary source of carbon for those corals. So while they may be living at a depth of 400 meters or 1,800 meters, they're in fact recording surface processes.
Right, so another connection from the surface of the ocean to the bottom of the sea floor, even a mile down.
Yes.
Okay, we're going to go back in and zoom in on these 3 small coral colonies. Thanks, Brendan, for that. That was great information.
My pleasure.
Brendan, while you were talking, I was just having a look here at a paper by Owen Sherwood and colleagues, and they didn't work with Bathypathes black coral, but they have got some measurements for Storopathes black coral, which is very closely related to the one here. And
Can
they
you
found
pan
an
up,
axial growth
please?
rate of approximately 1 to 1.5 centimeters per year. So pretty slow.
Say the growth rate again, please.
1.2 to 1.36 centimeters per year. That was the axial growth rate.
Okay.
Up the maximum 66 microns for the radial growth rate.
Yeah. So that's in line with what you'd expect. It's a little bit faster than like the Leopathes.
Okay, and then you can pan up.
Yeah, that's
and
pretty
move
good.
left. Get another Paramecia, so you can see several very small Paramecia. Actually, you see that small one in the front? Are we on full zoom? Looks to be a really small brittle star on that colony. And then we have a red colony in the back, maybe. You see that, Tim? The very small oafs on these Paramecia colonies?
Yes, I do. You often wonder when they acquire this associate, when does the brittle star attach itself to these corals?
And what are the
Indication that
cues?
happens early. Yeah, no idea what the cues are.
Some type of chemotaxis, maybe?
Yeah, co-pilot, actually, now that they're farther out, can you tilt those lights back to normal? Copy that.
Yeah, absolutely could be. You know, in many coral species, we only allow one, let's say, brittle star to live on them. So what do? How do they keep the other ones off?
Yeah,
If they do.
exactly.
I think we're still trying to
Yeah, you
identify
can
the
pull out.
patterns before we get to the process.
Pilot, you can pull out, and then to the upper left, there's a large black coral. It's dark red.
Can we go wide video? Great. Kyle, do you want me to bring these all the way up? Yeah, I think all the way up is the default. Okay.
Okay, and we're gonna move here to take a closer view of the black coral, and just our shore-based science team. Are there any other interesting species or features that you'd like to get closer zooms in on?
I'm good to move.
Yeah.
I'm good to move.
Okay. Yeah, sorry there. We, you know, sometimes when we come down and land at first, it's really exciting to see new species, and seen several small corals today. The Chrysogorgia, I don't believe we've seen yet on previous dives. In my experience in the deep Gulf of Mexico, the species of Chrysogorgia that exist there do occur a little deeper. I know there are some species that occur in shallower depths, but Scott, I don't know if you could add to that in this region, North Atlantic seamounts, if you also see most of the Chrysogorgids in deeper water.
Hi, Andy. New pilot.
Hi, Dave.
What would you like?
There is a black coral behind the fuzzy Chrysogorgia. We'd like an image of that, and then we can continue moving.
Auto stabilization dialed in. Am I anywhere close?
To the left, pan left.
Bringing my head left.
And it's coming into view. Dark red.
Dark red.
Yep.
Yes.
And we have Dave and the head pilot chair here.
Hi, Andrea. Sorry for delaying. I was waiting for you to finish speaking. Certainly, we see Chrysogorgia deep, very deep, 2,000 meters, deeper than 2,000 meters. But they can also be found up 400 or 500 meters and so on. So I think they would be found throughout that whole depth range.
So you just stirred up the sediment here.
I should also add, I think there's more than 36 species of Chrysogorgia. It's a very large genus, lots of different species. So not every species has that wide depth range, but the genus, that morphology of octocoral is quite common across the depth range.
Correct, and some species may be distributed in specific depth zones, and you know this may likely change with different regions as well. Especially in the North Atlantic, where temperatures are more uniformly cold, some species may have larger depth ranges compared to, say, in the Atlantic in the Gulf of Mexico or off the southeastern U.S., or even in the Caribbean Sea. It'll be interesting if we get to explore the Caribbean Sea in the future, if we're going to see the same types of corals there and distributed in certain depth ranges.
And that's a good question. And AJ actually had a question about that on IM earlier, if anybody had a handle on whether or not these corals might be susceptible to temperature changes or what kinds of temperature changes they could handle. One of the points to make is that, at these depths, you typically don't see large changes or even relatively small changes in temperature. So,
right, at these great depths.
Yeah.
Yeah.
So,
but on the upper continental slope in some
Yes.
regions, we'll find huge variations in temperature.
yes,
Well, huge being maybe 5 or 6 degrees in some
Yes.
cases.
yeah.
But you're right, at this deep depth, it's fairly uniform.
Temperature is pretty uniform.
I see what's going on.
And we're just trying to zoom in and get a image here of this black coral. Again, something new we haven't seen yet.
Did I just make a divot there? Is that what I'm doing? I'm tripping over the... I think it is. This is not going well. Stand by. More zoom, please. Let's hold that for a while. Hi, science.
Hello?
Hello. I got a brief from the previous navigator, but wanted to get a sense for the plan.
Nav, this is
Can you get up close? It looks like a black coral here, but this looks different to me than the other Bathypathes that I've seen. It could be a different species of Bathypathes.
We're going to go really slow like this.
Yes, we have been going very slow up this. We actually haven't moved in quite a while, so.
Got a grip on things now.
Okay.
This is a good view of the polyps. The polyps on these types of black corals are quite odd. They're really stretched out along a branch. And so you'll see there's a little mound, which is where the mouth is. There are 2 tentacles around that mouth, but then the other 4 of the total 6 tentacles of a single polyp are visibly away from the mouth. And it was originally thought that these were dimorphic polyps, but it turns out they're just really stretched out polyps with chambers within them that house eggs on the side.
That's interesting. That's great. Thank you, Dave. Okay, I think, is everybody happy here? Or is it time to move on? We did have a question from Shore. And Dave, maybe you have a handle, or the other people in the room here, about the current. When we first settled down, it didn't seem like there was much current, but we're seeing more visible movement when we stirred up that sediment.
Well, I'll tell you what I'll do is I will lift off very gently here. Let's come wide video, and we'll see which way we want to go.
We have a red crab under.
Sediment, unfortunately, is my own fault.
Yeah, and it's still hanging. We don't really have a strong current here.
Uh-oh.
All right, I think we're ready. We have another Cursogorgia in the back on top of this scarp. And we are headed up to waypoint 1. And our depth is 1768. Local time is 11:14. Today is the 16th of July, and we are surveying the east wall of Atlantis Canyon.
Rich, up slope. If you want to sidle over to your right a little bit, get kind of serious, but the vector is roughly 100 degrees. Got that? 100, copy that. Alright, and we'll maybe see what we see as we come over this rise before we start moving the ship.
Another Bathypathes. Is that the same one? Or did we just change direction? I looked down for a moment. I missed it. Hey, shoreside scientists, is there quite a bit of a lag today in the video? Just curious. We're moving over up a scarp here with the thin veneer of sediment.
Andrea, Scott, I'm not sure if the lag here is any different. It does seem a little bit slower to lose the pixelation than it has in previous days.
Okay. And we're just panning left, getting an overview of the seafloor. And we're looking east right now along the wall. Not a lot of conspicuous fauna, although when we stop and take a closer look, we do see higher colonization of very small individuals. Sometimes that's hard to see when we're far away or off the bottom. So having different camera views is very important, I think, to our work. Getting a good idea of the bottom on wide angle and maybe larger mobile fauna, mobile megafauna, and then getting close-up views is extremely important as well.
We'd like to request a ship move. 10 meters.
Yeah, we'll take a snap zoom in on that you're honing in on, Dave. You got it. We are at a depth of 1763. It's a bit of debris here. With some rock rubble at the base. Another black coral. Maybe the same genus of Bathypathes, but it does look different to me. And we call these black corals because their skeleton is black, proteinaceous material, and then the red that you're seeing are the actual living polyps, living tissue that covers the skeleton.
Andrea, this is Tim. Sometimes we see polychaetes wrapped around the base associated with these kinds of Bathypathes coral.
Okay, cool. We'll get a close-up view and see. Can we zoom in on base?
Get into the Zen zone here. The vehicle and the pilot calm down a little bit.
Is that a shrimp to the left of the lasers? Tether pulled Dave. Well, he pulled the ROV back, not Dave.
We have to appreciate yourself. I am the vehicle.
You are the vehicle. So
That's why it's
if Dave is getting pulled back, then that's all right.
looks like Sarah should be catching up soon. Yeah, I pulled the flight down.
So we're just going to get another zoom in on the base here to see if we observe polychaetes, type of worm living at the base. Often we find polychaete worms associated with this species of Bathypathes black coral. We're at a depth of 1764 meters, and local time is 11:20.
Thank you, Big. Okay, video, let's zoom. Light.
Another brittle star on the rock face. There's a sponge right there too in view. Another glass, encrusting glass sponge.
We've also seen amphipods on these branches before.
Are those extremely long polyps at the base? I've never really noticed that before.
Or branches.
It's very cool.
That's because I'm not touching it.
Curious to what Scott thinks about that.
Tilt up here on the side.
Yeah, these polyps seem quite a bit longer. There's more branches on this Bathypathes, and we were just imaging a Bathypathes just a little bit ago. Again, these are black corals defined by their black color of their skeleton. Often they have red tissue, although we also see white tissue on black corals. Red, pink, or orange, I should say. I'm not seeing any associates, are you guys?
No, we're not.
And then just pan right, and then we'll be ready to go.
Great, thank you for the look. It's always great to see these corals close up.
Yeah, definitely.
Polyps at the base are so interesting. They sort of shroud the base, and we've seen those before, even
Yeah, Bridge, we'd like to
longer
test,
than these,
speed of that
extending
move,
beyond the
10
base.
meters, 100 degrees.
I don't know if I've ever noticed before.
Thank you, Bridge. Here, stars.
Another sponge,
Ah,
another glass sponge.
quality of time. Let's
Can we get a zoom on the field, just a partial zoom while we're here?
There's two Okeacanthid type varieties. And then after that, just we can pan right too to get a view of the sponge.
coming up. Back in.
Andrew, is that a crinoid too, just to the lower right? Looks like a stalked crinoid.
Can we pan right too and down?
Right down. Okay. How far we need to go?
Don't know yet. And down. Looking for a crinoid, possibly. Can we keep going down? Yep, right there. We have a stalked crinoid.
I didn't do that, Brian.
You know, Dr. Charles Messing was really interested in finding out more about crinoids in this region, noting a lack of information in this region on crinoids. And we actually have a few people interested in the distributions. I mean, we all are interested in the distributions of these species to further refine models and understand the distribution of these species across ocean basins even. Or whether the same species occur or sister species, closely related species, understanding their distributional range. Yesterday, I know we had some folks excited about the king crab observations, noting that it was the
Another little
most
forest
northern
layer.
most sightings of king crabs.
a hole right above. So, push focus into the hole.
So we're going to get a close-up image of this crinoid.
We got any more zoom?
Yeah, Dr. Messing sent us some keys for crinoids to help identify them. Not easy at all, but we'll try to put some tentative names on these. We can look it up later.
Good, Rich.
Yeah, exactly.
Thank you, Rich. Andy, I was quite startled to see a crinoid running once.
You saw one running?
Running. A crab. Skittering across the bottom in a spider-like fashion.
In a spider-like fashion. Wow, that's very cool.
And,
Alternating the arms to move across the bottom.
yeah, I was running, I guess. Like a spider.
It wasn't swimming.
No, it was pulling itself across the bottom.
Very
Yes,
cool.
and, I mentioned that to somebody and they didn't believe me. So
I believe you.
is this known?
Let's see if anybody knows. Has anybody else observed that before? A crinoid using its arms to run?
Actually, it was the hold fast. It was the bottom end.
Oh, it was a hold fast.
Yeah,
Really?
it was a red one that had a hold fast that looked something like arms, claw-like, whatever. But it was,
Huh.
it was going very quickly. I think we can come out video.
Yep, we can come out. And Brendan Rourke is going to take over here for a little bit.
All right.
So Dave Wright was just asking that if anybody out there had seen any other crinoids walking or running on the bottom of the seafloor, not swimming. He did not observe this swimming, but he actually observed, Bob Carney. He's seen that as well. Crinoids running and looking like spiders. And
Yeah, and their arms sent backwards like the hair in the wind.
all right.
Got it. Okay, so we are at a depth of 1762 meters, and we are making our way towards waypoint 1.
Hey, Andrew, it's Jason. I'm back. So you can do geology again now.
All right, Jason, welcome back. We've been doing a lot of biology, so we'll save the geology for you now that you're back. And we have,
We have a rock with a coral.
yeah. This is one of the bigger coral specimens that we've seen so far today, so we might as well take a zoom in on that, please, when you get a chance.
That looks like a bamboo coral coming up.
Yes, I would agree with that.
But you always agree when I say bamboo.
Hence my biases. We've already talked about that.
Yeah, but it's a new
The
day.
crinoid on this bamboo.
Sorry, say that again, Scott?
Yeah, sorry.
I said, yeah, but it's a new day.
Yeah.
We're looking for filler.
There's another coral in front, and a crinoid on the larger bamboo. Crinoid down to the right a little bit.
Okay, you can zoom in on, get a close-up shot of the coral, and then there's a crinoid down below and another coral down below. So,
Hey Dave, just an update. We've got a delta of 15 and camera angle 33. Okay, I've got time. A bit of a zoom for this video, and I'm going to rise, hold that, and work my way up.
then at some point, if you can pass those lasers close to the base, that'd be great, but only if it's easy. Perfect.
There's 10 centimeters. Pretty fine, right?
Yeah, this is by far the biggest bamboo we've seen in almost, I think, all crews so far. So,
I agree, Brendan.
Give me a little more zoom, please, video.
and again, the depth is 1759 meters.
Let's come out for a second. I'm going to work over and get a better angle on the holdfast.
You're good on the hold fast, I think. You can do the one, the coral down in front as well, please.
Okay, coming left. Zoom back in.
Looks like another smaller version of the same one, maybe not branched yet.
Yeah, it looks exactly like the same, so, but definitely smaller. You can see the nodes through the tissue at the base. I count at least 2 nodes easily identifiable. Can you get a zoom in on it
Interesting
as
that
well?
both of these, the larvae appear to have settled on the vertical surface and curl outwards.
Yes. Perfect. And if you pan right and up, you'll see pan right.
Yeah, I think there may be a soft coral in the crack of the rock there.
Yeah, that's what I'm trying to get him down to. So pan down, pilot, please.
What was the feature description?
It looked purple.
Out slow. Video. Out slow. Hold that.
There you go. You see it?
Oh, yeah. Okay, we'll zoom back in. There. Okay, get that close to the time to go here.
Okay. Is there anything else shoreside that you want to see here?
So pilot, you should have time. Series won't swing. You should be about that.
I think okay.
There's more stuff on the other side of this rock here that's
Yep.
interesting.
Thanks, Nav.
I'm actually caught up with them right now. So the first part of it was about 4 minutes long. Basically, they zoomed in, they zoomed out, and they zoomed in at the base of it. So this is basically just the second part of it.
Brendan, is that a chronoid there in front of us, or is that a Metallogorgia coral?
Pilot, can you do a zoom on the... Yes, please.
So that.
Put it in there.
Hey, that's a Metallogorgia.
Do you want more imaging
Yeah.
of it?
Yes, please.
Juvenile Metallogorgia. It's got the branches still
All
about
right,
midway up.
pilot, you're here in the chatter, so yes, we will require some additional imaging of that one, please.
And it should have a central ophiuroid. I think I can see it now.
Yes, so we'll need framed and then zoom in on the top part and then zoom in on the ophiuroid halfway up its stalk, please. And I'm going to type in a lat long for this location that we just dropped a DVL target on.
You know, I think this is really interesting. If I'm not mistaken, this is the first metallogorgia that we've seen this far north, not on a seamount. I don't know that we've seen any on the slope, and maybe Les can correct me if I'm wrong. I don't believe they've seen these further north up off Newfoundland. I think the furthest north I've seen these on the continental shelf, slope, is around Bahamas, Little Abaco Canyon, Florida area.
Okay, so
I would agree with that, Scott.
Auto XY with a current, with a little bias to keep the thrusters spinning. In this terrain, it causes this sort of thing to happen. Right, and they don't have much current today. Yeah.
so we had a little bit of a problem with the auto positioning that got a little bit skewed because of the varied bottom terrain. Kicked up a little bit of sediment, so we'll give it a minute or so to settle out and go back in and try get some more imaging of that.
When it's working well, it does a better job than a pilot, and when it's not working well, this is what happens. So we just have to learn more about appropriate use for it.
You guys are doing good. That happens.
Let me think I can brace myself ever so gently. We've made some really good macros here. Just need to gingerly approach. Let's put a little more zoom in it and see if I can load it from here on. Just hold that right there for a second. I think these new branches are of interest. A little more zoom, please. Just going to work my way up the stack here in a second.
That's great work.
A little bit more zoom. Try to see the sure.
Does anyone think that there's more than one opioid on this?
Looking for centers.
No, single.
It's they're pretty jammed up in there. Would be that either. Okay, it's staying there. I'll set it off.
There might be a second one on top.
Yeah, up there.
Yeah.
I think that's just an arm extended.
Yeah, I agree.
Yeah, you can see going towards the bottom. The arm extends quite far. So this looks like it's just one.
Yeah, I've got my arm up against the rock to steady myself here.
You can do a full zoom if you want to, if you're steady enough.
That is maximum zoom. Sorry.
Okay. Thanks, video. All right, whenever you're good, we're good.
Okay, come live rolling.
Yeah. Is there anything else that you want to focus on here, shoreside?
I think we're okay.
Okay, let's keep moving. Let's keep moving, pilot. Thanks.
Looks like there might be a hard target about 35 meters ahead, Dave. I see it. 29. I've got more of it in my sonar. So whole, more of the same. Yeah. Looking back. It's the face I braced against. And coils away.
Okay. So that's a good view that sort of shows again that you know, we're looking at certainly where we're seeing more of the deep sea corals, something much closer to a vertical face. And the biggest bamboos that we saw were sort of at the top of that vertical face. Another small sediment patch, and then back to the large slab of hard substrate, the calcium carbonate cemented rock material.
Yeah, bridge requesting a ship move. 15 meters, 100 degrees, 0.2 knots.
And you can go in and take a look at that coral that's coming into view, pilot.
Copy that. Thank you, Bridge. Dave, I believe we're headed a little to the starboard of our current ship heading. I'm moving left. Yeah, that should situate us this way. Bring myself back. Let's get a zoom on this, please, Roland. Get an RD on this.
Looks like another bamboo coral.
More zoom. Sure looks like it.
Yep. If you can walk those lasers down to the base, that'd be great.
Zoom out just a hair. More.
Yeah, large expanse on the base. Great, thank you. And then if you can just zoom out a little bit and then get a partial zoom on the rubble at the base, please.
Okay, a little bit more zoom in. Got somebody's living on this like cyanide on the back side of that rubble.
Yeah.
And
Bunch of sea stars.
stuff.
Yeah.
Little stars.
Can you zoom in on that, what looks like a cup coral right there where your lasers are?
Yeah, zoom on. Okay.
Can't tell what that is from here, but looks like a cup coral. Very small. All right, we're good, pilot.
Go wide, Roland. I've got the side off to my left here.
Okay, let's see. Maybe you can work your way up this slab. Take a quick focus on that.
Yeah, that takes me away from our direction of advance. I can pop up and give you a quick look as I move to my left.
That works.
Snap zoom,
Roll? I don't think we're in any risk, but just remember there's that rise behind you.
Yeah, it looks like another Bathypathes. Just tad up, perfect.
Come on. Some cereals is starting to close. Science, there was a purple stalk underneath that first pink one that we got. I don't know if anybody saw that or if we recorded it yet. Go, Bridge. Thank you, Bridge.
Under.
Bottom right. Okay, head and change to 300. Got the frame.
Okay.
It's about 8 centimeters long.
Let's keep going up, pilot. Do a zoom on that one when you get a chance, but quick zoom's fine.
All that zoom. Coming right.
Another black coral, Bathypathes.
More zoom. Hold that.
And that gives you a good example of where they get their name from is the skeleton. And it's pretty common to see a pretty large holdfast the bigger the corals get, or these black corals get, in order to hold on.
Can we zoom on the opioids there? Looks like opioids. Is that what those are? On the. Oh.
This is an interesting one because it's much more complexly branched than the others we've been seeing, and is likely a different species.
Yes.
Yeah, I agree with that.
We're at the end of the ROV's tether, so pilot is going to have to come back around to his original heading. Are you at your limit, pilot?
Yeah, I'm stretching the tether
Okay.
here, but we are headed that way.
Okay.
We believe to have a shake in his head. So things will get better. We're just a little deeper today, so things
take a little bit longer.
slower.
If we're heading this way, then let's plan on, you see the sponge that just went out of view, and then what's at the very top of your screen right now, and then the bamboo coral, if we can get snap zooms on, and then maybe the black coral, if we can get snap zooms on all four of those as we go by, that'd be great.
Set up for some good light, and I see bringing our delta down a little, so I'm more tethered to me. Tony's giving me some leash to work with here. Okay, I'm at 14 right now. That's right.
Okay, so we are basically pointed directly at the wall, and our heading is 150 degrees. Our depth is 1752, and we're going to image these sponges and corals on this wall.
Flacking real fast. I see something in the center.
Yep.
It's a bit purple. Inches up to it.
Can't tell what that is. Huh.
Sorry.
Tim is wondering if it's a shrimp, but I can't tell.
From video's perspective, it looks like a shrimp.
Yeah. Yeah, there you can see sort of like its eye and then up its body.
Okay, I'm just gonna rise up a little bit here.
All right, we're good if you're good.
Spin out a little bit, roll, and I'll pick up the next one. Okay, just next to it. Just
Yeah, do those two as well, right next to it,
come
please.
back in, rolling. The yellow one on the rock there, I'm working on that. Pushing in, following you. Yeah.
Looks like there might be, yeah, it looks like, well,
Let's hold it right there. Now we get a two shot. Nice.
that looks like a cup coral. You can do you can do zooms on both of those if you want, real quick.
Rock in on this guy. Maximum.
Okay. That's good for science, and then
Just a little tether bob going out. Going out. That's alright, hold it. I'll get it back. Back in, pushing in. Wait for it. There you go, you know?
all right.
Caution. Yeah, great for testing another ship move. 5 meters. Small depth of field on 100 degrees.
All right, we're good on that, pilot. Thank you.
Full line. Video swapping out.
Okay.
Respect. There's another bigger coral above you.
Yeah, so we're looking at, do you remember where that, it looked like a sponge was? There it is, right to your right.
Yeah.
Let's take a look at that. So we're looking at a bamboo, at least one black, relatively large sponge, a Parametheus.
Co-pilot, I'm gonna restart the sonar. Okay, thanks, man.
A cup coral,
Just hold it right
a number of smaller sponges. You can zoom in further on that whenever you want.
Okay, cut in smaller first here. Got some sediment in there.
Yeah, it's got a fair bit of sediment in there. Nice.
Yeah, keep going. Thank you, Bridge.
Nav, can we drop another target, DSC2, please?
Copy that, science. DSC2.
Thanks, Nav.
Come out now. Some other goodies on the wall here. Come out, does some more. More options. Full wide, actually. I'm
Shoreside, is there anything else that you want to see? Another bamboo off to the right.
There's a call here to try to see if we can't see the base of that sponge. We're just looking at, so it just comes from the side a little bit. Yeah.
I hear Tim faint the base of something.
You want the base of the sponge, Tim?
Yeah, that would be great.
Okay. Sponge
You heard
base.
correctly, pilot.
Okay, let's start slow.
Okay.
Let's hold right there. Get a little more zoom, please. There we go.
Great, thank you guys.
All right.
Thanks, pilot. Anything else shoreside? I'm trying to type you in a lat long for that DVL target real quick before we move.
You want to brief me my
Yeah, I think we're done here.
I don't think so. 6-5 north.
All right, it's Andrea. I'm back. Just catching up here. Brendan is updating a lot and long. I see Chrysogorgia in view and a sponge, and there's a black coral and a bamboo. Have we gotten images of all yet? Or do we need images of all?
I think we've gotten images of everything that's been asked for. We've gotten some images, some earlier images of both the black and the bamboo. So I've been focusing more on doing quick zooms to identify that they're what species they are, or to get a good image of them, but making an effort to move up towards waypoint 1, which is still a ways away.
Okay. So did we did get an image of bamboo, quick image of the bamboo?
Yes.
Okay. All right, so we're moving on to waypoint 1.
I think we did anyways. So
Is that true,
We
shoreside?
didn't have the one here, but we did get good views earlier.
yeah,
Okay. So same species of bamboo here.
yeah. So, yeah, and this is a big, huge rock face here. So we're definitely starting to see the targets that we expected based upon Brian's estimations of slope. So
bridge now?
this should continue for a little while.
Just checking to see if we're moving at all or we stop.
Depth is 1553.
17.
Sorry,
Correct.
1753.
Thank you.
Okay, so moving on up, slope to waypoint 1. Who's sitting there?
Bobby,
Hey, Bobby.
finally have settled on shifts for pilot. So I have a feeling I will be your pilot for the remainder of the trip.
For the remainder of the trip,
Well, for afternoon.
for the afternoon shift.
We're on the wish days. for the remainder. That's it.
Did we get an image of this bamboo coral? Or the species of bamboo?
No, I don't think we saw this one yet. We were moving down
Okay,
this
can
way.
we snap zoom on that frame coral?
Yeah, I can't tell at this distance if it's the same one we imaged earlier, or the same species. I mean, we haven't seen this particular colony.
Quiet video. That was partial.
Good, hold. Is that a chirostylid?
Yes, I think it is. Yeah,
Okay,
I'd like to image that.
and then when you're ready, zoom crab right. in the coral.
Okay. Right and down.
Yep,
of it here.
yep. Getting close to your lasers.
Go ahead, video.
Oh no, not a crab, anemone,
Oh, anemone. And a barnacle.
a barnacle. Is that
Oh,
a?
I'm sorry, that's corals.
Is that a cup coral?
It looks like a cup coral to me.
The one below for
I
sure.
don't think you guys are hearing me. Is that possible? Maybe real delay.
Tim, we are hearing you.
Great. So I see 3 cup corals and supposed to be a barnacle.
Yeah,
I agree.
I agree too. That's the first time I've ever seen that.
Yeah, me too. Cup coral settling on a.
You notice how
You can see
thick
they're
that
settled
part
with
of
it.
the branches there, just to the upper left of those cup corals, and I wonder if there's been recalcification over a dead part of the colony.
Yeah.
It's sort of devoid of polyps there too. I think you're right, Scott.
And a little bit of an accumulation of sediment on that branch as well.
Can we go up one branch and look? There's a crinoid up there too.
Okay,
just
can you
above the
pan?
lasers.
Yep, I got it. Pan up.
Okay, I won't be able to get in.
Just above your lasers, to the right of the coral, there's a crinoid on the wall. Is it on the branch?
Is it on the wall or coral?
I guess it's on the coral. Just pan in, partial.
Do you have just a general?
Yep, to the left of your lasers and down just a bit.
Okay. Video, let's go partial. There's one in, see.
Yep, and it's in your lasers. Yep.
Yep.
And there's a shrimp. Yep.
Well, actually, it's a little easier for us up here if you give just a general direction as opposed to a pan left, pan right type stuff.
Oh, okay.
'Cause sometimes it's easier for us to change our altitude as far as pan.
Okay.
Instead of panning the camera.
Okay.
Other
Copy.
stuff.
And so can you zoom into the? Yep. So it's growing on the coral, and there's a small red shrimp. And we're getting an image of a crinoid.
Looks like the coral's been injured at some point, doesn't it?
Yeah, there's no coral tissue where the crinoid is attached.
Yeah, you just seen the skeleton there, the end of the skeleton.
Yeah. Which would then leave, if the crinoid moves, then that would leave open the opportunity for something to settle on, which is probably what we saw with the cup corals.
Oh, sorry about that video. Come on, Divine.
Yeah, science. I think we're good here with that. Would you like to see any more close-ups of the crinoid or branching
think we're
pattern or
okay.
anything? Okay.
Would it be possible in a different part of this colony to get a view of a branching point?
Yep.
To see if we can see if there's a node at the branching point?
Agreed. Okay, so
Yeah, video. I'll watch it stand by one and we'll see if we can
okay,
do,
maybe on the left of that coral.
right, yeah.
So for those listening while we're setting up for the shot, on the internet feed, one of the reasons that we're interested in bamboo corals in particular from a paleoceanography standpoint is that they have both of a gorgonin node and a carbonate or a calcium carbonate internode. And as I said earlier, the gorgonin nodes are essentially recording the carbon signature
So the one
from
sticking out
the surface ocean,
towards us on
and
the top.
yet the carbon The carbonate
Above your
internodes
lasers.
are
Above your
recording
lasers.
a signature
Yep,
from
right in
the
there.
depth at which the coral is living. So basically, when you're looking at a bamboo coral, you have a archive or a recorder of both the depth at which the coral is looking at or living at and a
Roger.
record of what's happening in the surface waters in the same specimen. So from a paleoceanography standpoint, that's a really interesting opportunity to reconstruct both surface conditions and deep water conditions.
That's a good view. I can see nodes on the right side, but not at the branching point, so this would make this more likely a keratoisis and not a jasonisis. Thank you.
Okay.
Okay, thank you, pilots. You can come wide.
All right, video, come on. Yep, that's fine.
Okay, I think we can continue our move.
Now, what's our move going to be? Well, I was informed to do very small hops, so we don't know what this face is like. Alright, still a pretty good hit, so if you want to just come up a little to see if it continues.
A few more glass sponges here.
I did probably 10 meters. Can you tell us what our bearing would be? 100, because we've been moving 100. Alright.
Some staining on the wall.
Jeff, let's get you to turn left a little bit to do that.
Headed for lunch? Okay. Yeah. Did we get a view of the sponge in the center? Okay.
Yeah, we did. Yeah, we worked all of those. The bamboo, yellow one, the sponge. There's a couple of cup corals around there.
Okay. Just getting a briefing from Brendan here.
This is good wall.
Can we get a quick zoom on the hot pink in the center? Two anemones. And a shrimp on the bottom left.
1, 2, 3. Testing. 1, 2, 3.
Or is that a coral? Huh. A coral. Sea star.
Does that look like a sea pen?
I'm thinking, yep, that's what I was putting in the vent log. It looked like that sea pen that we saw on the sediment earlier. Okay. Thank you, Bobby. Good. Yes, thank you. Maybe it'll turn for us again.
Testing 1, 2, 3. Testing 1, 2, 3.
Really good close-up view of the polyps.
So this looks like a sea pen that is on a hard bottom instead of a sedimented bottom. You can see the holdfast there quite well. Gary Williams and colleagues have recently written a paper describing what they call rock sea pens. And we did discover a new species down off of Florida, which looked a little bit like this grossly.
Very interesting, and so we saw a sea pen that was similar to this, but thinking that it might be a different species. Earlier, the one that turned to our view, giving us that great close-up footage. So this species is a species of Cophobelemnon, named Cophobelemnon francii, named after Scott France, who was just talking to us online. Species of sea pen associated more often with hard substrates. Very cool. And this was originally described from specimens collected off of Florida. Okay, another bamboo coral. That's interesting. Is that just hanging off the wall? If we can frame coral. I feel like I'm getting an echo. No. Okay. Another bamboo. Is that 2 bases at the bottom? Or 1 base, 2 branches? Several branching points. But 1 hold fast. Cup coral below, it looks like Javania. Okay. And do we see any associates on this? Get a better image of? It's a fairly wide, it appears to be a keratoisis. I think it's branching from the white parts, the nodes.
I turned my head for a second. Can we see polyps again?
Those zoanthids? Zoanthids?
What you're doing, Bobby, is good right now if you could hold. And thanks.
The darker parts there with the tendrils coming off of them. See those wispy tendrils?
If you can move right.
Nice view of a node there.
I'm not sure. You're great view of the polyps on the right. And question whether the ones on the left are zoanthids or those.
Yeah, they're really odd looking. They don't quite look like zoanthids from this view, but
They almost look like the bamboo coral polyps, just of a different color. They resemble
I've
that
never
to me.
seen that before. The filaments may be, filaments of mucus.
Okay, we can come out. All right, great, thank you. Anything else here, shoreside?
No, I think we're okay.
Okay, thanks. Okay, let's continue on, Bobby. Looks like another Chrysogorgia. The zoom on the brittle star on the top. Again, we're seeing that one species on the seafloor at the base of corals. Actually, a couple in view, and a different brittle star on the coral.
This looks like Metallogorgia to me. I don't know if we can come from the side
Oh,
after this. Oh, great
maybe.
shot of
That's
the
a
opioid.
Metallogorgia.
Yep, from the side, yeah. Can we, after we film this, get an image of it from the side?
Yeah, I want to get
And
a good
these-
zoom on that disc. It's great.
And this species of octocoral often have a specific associate, a specific brittle star associate. We're at a depth of 1743, and our temperature is 3.7 degrees C. It's a large white sponge off on the right.
Andrew, we just saw a very large sponge off to the right at the same level. Just pan right, you would see a very large sponge.
Yep, I just saw that. Is that genus? Do we have a genus on that? Is that Pharea? And we're just moving this up to get a better image of the Metallogorgia from the side. Then we can go take a closer look at the sponge. Oh, this is great. This is good. Keep it in frame just like that. And we're just taking a close-up image here of Metallogorgia with a brittle star inhabiting the coral. The brittle star may be Ophiocreus oedipus, which often is found in this species of coral. So another example of a specific interaction between a faunal associate, in this case a brittle star, with an octocoral. Okay, are we good here? I'm thinking we can go take a closer look at that sponge. Okay, you can come out and move right towards the sponge.
The whip down there, the bottom left.
Bottom right, the sponge.
Bottom, no, bottom left, like a cladoisis or something.
Keratoisis, bottom left.
Look in the HD2 view. You'll
Yep.
see a whip coming off to the side there, white. I don't think we've seen that before. Or did we?
Yeah, pan, yeah, down. Sorry, I've been saying pan all morning. I'm trying to stop saying it. The one on the left, Tim, coming into view?
Just coming to view now, right?
Yeah,
And
on the left, right?
ROV on the left.
Yeah. Yeah, so we're in between. The sponge is on the right, that coral is on the left. I'm thinking we can go take a look at the sponge real quick, get a zoom in on that, and then we can come back across, because we're headed that way, correct?
Correct. Thank you, Andrea. Yeah, I don't want
Okay.
to interrupt what you were doing, but I just
Oh,
saw it
yep.
there and thought we should note it. Yeah.
Definitely. And we can take a closer look at it as well. Yeah. Okay, the sponge. Take a frame the sponge and get some imagery of that, and then we'll move back to the left. Oh, what's this? Oh, and there's a Paramecia colony on the right. Okay, close up here. The sponge looks a little different to me. A lot more structure to it. Looks like a glass sponge. Hexactinellid. We'll see. Is that a brittle star in the back? Yeah, the sponge has a lot more texture. That's a good word. I'm gonna steal that word. Few brittle stars. You can see its arm peeking out around the back of the sponge. Two of the arms. And then if you move down a little bit before you come out, oh, okay. There's another star in the middle. It's, yep, by the lasers, yep. Is that a squat lobster on the left? You see that, Tim? No, not a squat lobster. And just getting some
Yeah, it
close-up
looks like
imagery.
it.
No. You can only see it lodged in the sponge. And then if you, there's maybe a crab on the left, and that's good. Not sure if we can get to that. It's.
Yeah, I
Can you
don't
see
think
where I'm
so.
looking above the lasers? Yep,
I do.
right there.
Yeah, right now. Yeah, zoom right now might catch some of it, but
Yep, squat lobster. Very cool.
wow, nice shot.
I've never seen this species of squat lobster. How about you, Tim? It's got pink eyes, pink orange eyes.
Well, it looks like a munidopsis of some sort, but that's a big group. Yeah, I don't know either. Not without.
I'm not sure yet. It could be a juvenile, but there are some adult species of squat lobsters that are fairly small that we haven't found to be large sizes. I would say an adult squat lobster, likely.
Yeah, I agree.
All right, Tim, are we satisfied with that?
Great, thank you.
Okay. All right, let's continue moving back towards the left. There'll be a coral sticking out of the bottom that's like just one long stick-like shape. We can take a frame of that and keep going. Not yet. It's past the pink coral we were viewing. It'll be more on the left if we continue along the slope. We're currently at a depth of 1743 and our temperature is 3.7 degrees. Local time is 12:27. Still moving to waypoint 1. I just want to bring to everybody's attention, oh, go ahead, Tim.
Never mind. Photos on the metallic gorge again, and that's it right there, right? At the left, upper left of our screen. Upper right of our screen. That's metallic gorge where we were, I believe.
Correct. Yep. And then we're moving to the Lepidopsis, and I think we should think about getting underway and covering a little more ground. This has been a great dive so far. We've found at least, again, right around 10 coral species, I believe, or possibly more than that. Maybe up to 12 or 15 now. Different species of bamboo corals and Chrysogorgia, Metallogorgia. We haven't seen those corals on previous dives. Different sea pens than we have observed so far. And several sponges. We're picking up new species here. We are deeper than we have been all week. We're also in a different part of Atlantis Canyon. Maybe a couple squat lobsters.
Different.
Yeah, go ahead, Tim. What were you going to say? Each dive's been different?
No, I'm just going to remark that is the case. I mean, every day has been a different suite of species we've seen and habitats. It's been pretty amazing.
We're viewing a bamboo coral, maybe a Lepidopsis.
I think seeing the tip of it's really important to see whether it's branched and forked or not at the end. Got a little twist at the end there. It's had some damage.
Oh yeah, there's some damage at the very top there. Or just not growing. It's colonized by some hydroids. And you can see where there's no live tissue or the white tissue covering.
I think ultimately you have to have a sample of this to figure out what it really is. I think the morphology is very enough. You really need to sample it.
Yeah, and I think
So
that's a good point to bring up here now again, is that, you know, we're doing our best with imagery, but it's always good to have samples to confirm what we're seeing, and sometimes morphologies can change from location to location, and even within microhabitats, right? So sometimes we'll see different morphologies of the same species depending upon the location on a certain rock. We saw that with Paragorgia the other day, and often just a piece of the coral will help with identification. Okay, I think this is good, Dave. Tim, do you have any insights today on comparisons to other dives, or?
yeah, well, I think this is strikingly different to what we've seen in all previous dives here in the canyons and on this expedition. I think what I'm seeing here more resembles what we're used to seeing on the seamounts, the New England seamounts, cornerized seamounts. Seeing Metallogorgia coral here, bamboo corals here, even the Parameciids. You know, early in the dive we have the different kinds of ophiuroids, which we had not seen on this cruise before, and we don't really see those types on the seamounts where we've been because they've also not been really sedimented dives per se. But it's just strikingly different from any other dive we've had, and you know, when you go, maybe it's because we're deeper, and a lot of our seamount dives have been deeper. I'm not sure if other canyon dives that other groups have done this year and last year have been this deep, but it could be. We know that depth causes a strong zonation in the composition of fauna. You see that on almost every deep sea environment that we go to, whether seamounts or gulfs or even mid-ocean ridges. So, you know, just another example of how we know so little, but keep trying to understand the patterns. So I'm really, it's striking, you know, there's parts of the Gulf of Mexico that sort of look like this, that have Metallogorgia and the bamboos, like we're seeing here. But again, that's a deeper depth. So I think the lesson today may be, you know, there may be strong depth zonation in these canyons.
Tim, your audio is about 40% too low.
Yeah, I was going to say some of our dives we've conducted in the Gulf and DeSoto Canyon, we're seeing the same, maybe not the exact same species, but definitely the same genera. Whereas we get into the shallower depths, we pick up different species.
The Taylor Holiday, just let remember we've had a couple of deep camber toes and canyons north of here. That, so we did not see these fauna. So, you know, we've seen a lot of patchiness too. That's been a theme of these dives into these canyons. We've seen a lot of patchiness and colonization, and so patchiness may vary on a local scale and a regional scale. And this exploration we're doing now is really going to help us start to get a hold on that, at least form really constructive hypotheses, right, for going after these kinds of questions.
Right. And we're picking up that same species of sponge that we were viewing earlier. I think it may be. Yep. Yep. Let's move on. Are we moving along the slope, or are we headed up over the slope? Okay. I think we're. That's an interesting ledge. Let's have a look underneath this ledge real quick. Snap zoom. Maybe a Bursingid or a crinoid.
Another Metallogorgia.
Yeah, another Metallogorgia there.
I think that's the one we just saw. I think we're coming back over it.
What do you think of that, Andrew? Is that true? I was wondering.
The sponge we just saw was the same. So yeah, I think that was the same Metallogorgia. Yes, where we are. Yeah, let's move up. A couple cup corals. Can you do a snap zoom on that ledge right there? That's new, right where your lasers are. No, mostly dead. Three brittle stars here on the bottom. Looking at a rock face that's been heavily bored out. Call this bioerosion. Yeah, there's a patch of dead cup corals there. Actually, looks to be all of them are dead, covered in hydroids. Oh, what's that green? Can we see the green on the? Yep. There might be one live, a recently dead cup coral. No, they look all dead to me. I don't know, we saw this earlier. It was questioned whether this was a tunicate or a sponge. Sometimes it's difficult to tell just from imagery alone. You don't have any other ideas or educated guesses? Several worm tubes. Okay, we can come out, Dave, and keep going. Our depth is 1743, local time 1237. Just a reminder, we're still moving to waypoint 1. Not a lot of ledges and overhangs here again. I think we were mentioning earlier that this was more resembled more of the shallower dive here in Atlantis Canyon than Hydrographer Canyon, where we were over the weekend. Yeah, can we get a zoom in on that? Looks like a sponge here, so this is probably a sponge. Couple of Paramericia colonies in the upper right. This is different than we were just looking at, though. So we're zooming in on a yellow sponge. Have the lasers at the base. It's about 10 centimeters, 12 centimeters wide. And we're definitely looking at a glass sponge here. I'm gonna ask Shirley Pamponi, are you on the party line? Okay. Several Paramaricia colonies, each with an ophiuroid.
I'm here. Did you have a question?
Hi Shirley, how are you today?
I'm good. How are you?
I'm well. We keep talking about sponges and glass sponges, and I was wondering if you could tell our viewers if there's a quick way to tell a difference between a glass sponge and other sponges that they may be more familiar seeing in shallow waters. Is there a difference in their spicules?
Yeah, there's definitely a difference in the spicules, but of course, unless you have a sample of it, you're not going to be able to tell. Most of the kind of real translucent sponges that you're seeing, and the ones for sure in deeper water, like we're in right now, are going to be hexactinellid sponges, and they're going to be more, like translucent sponges. You'll see, as they get denser, and you see demo sponges. But I've seen a couple that I think might be demo sponges, but they were tiny, tiny, tiny.
And they aren't as easily transparent, I guess, as the glass sponges.
Sorry, I didn't copy your last.
Desmo sponges are more opaque than glass sponges.
Yeah, yes, correct. And usually a lot denser too. Although having said that, Gush sponge could potentially be a demo sponge, but it looked to me like a hexactinellid. We're going to put in a new code too, SPO-HEX.
Great. Thank you, Shirley Pomponi. It's great to have you today at the Interspace Center.
It's great to be here.
Okay, and we moved upslope a little bit. We're at a depth of 1728. And our time is 12:42. Looks like we're moving more on top of this feature here. Approaching waypoint 1. Was that sediment cloud from us? So Dave just talked about Little Herc, and Little Hercules was the ROV on the ship prior to the deep D2 ROV. Is this the second quote-unquote expedition with the D2? Thanks for that, Dave. Several Paramaricia colonies on the top. I'm seeing at least 10 in view. More in the background. There's two the bottom of the video. Looks like we have some more outcrops up ahead. And we're approaching waypoint 1. Cutthroat eel. Yeah, can we? Okay, good. I was just going to ask that.
Stuff like turtle coming over the brow of the hill, and you can see a bunch of wading and no exciting squeals heard from participants. But I'll wander here while I see those flags come.
Thanks, Dave. I'm just updating the lat lawn for folks back on shore, so I'm not looking at the video right now.
I'll let you know if there's anything noteworthy.
Now we're seeing Paramecia colonies here about the same size, although I bet if we look in closer, we may see smaller recruits as well. I think earlier in the dive, as we zoomed in on a few corals, you could see small colonies just a few centimeters tall off the seafloor. Right, and I agree with that, Scott, too. We're not seeing very large colonies. And I have a question for you regarding that, and Tim and others on the seamounts that have worked at deep depths. We noted in the Gulf of Mexico no very large colonies at deeper depths, around 1,800 to 2,400 meters. Paramecia weren't as large. And we saw sizes that were all about the same as well.
Yeah, occasionally we'd see some larger ones. I think you guys have paid much more attention to size, frequency, and depth and so on, so I can't answer the specific question about whether they achieve a certain size at a certain depth. But, you know, every once in a while we'd find a bigger colony, but on order, yeah, usually they're about the same size. I don't know if they're reaching a maximum limit, if there's something to do with the currents or the food input changes over time and they slow down in their growth, or what it is. Kind of interesting.
Exactly, and I should note here that, and Brendan is still away and he could talk more about this, but size does not always equate to age with corals. Sometimes they may stop growing or level out or reach an asymptote with their growth. And so two corals of the same size could be very different ages.
Right, you have to take into account the lateral growth. You know, some may have a lot more branches or branches going out farther to the sides and may not necessarily be taller, but then some you find are much taller and kind of narrow. It's rather interesting. Lots to learn here.
Exactly. Okay, we just passed over a white urchin. A few more of those white ophiuroids on the sediment on the seafloor. Pretty abundant here. And we're coming up across this more outcrop looking area. We're looking due south. Okay. Uh-huh. Correct.
How do we want to do that? Do we want to go along contours up or up and along? What's your goal?
To move to waypoint 2. What can we zoom out a little? Yeah, can we do a direct beeline there? Or moving along the contours and up as possible? Okay, let's do that. So we're about ready to move over waypoint 1, which again, to jog your memory, it's our old waypoint 4. So we landed on the bottom at about 9:30, and really have just been working this wall. We've found several new coral species here today. Not new species, but new for this expedition so far. So really exciting. We wanted to get really good imagery, and besides the corals, also sponges. Sponge diversity seemed a bit higher today, more glass sponges and different ones than we've observed on previous dives. And we're at a depth of 1723. Oh, really?
Well, this is Tim. Sorry, this is Tim. And just to comment a bit about what you and Scott were talking about in terms of the size of Pammersians we've been seeing, and I haven't been doing a size frequency analysis either, so this is just sort of anecdotally what I feel like the impression I'm getting is that we don't have the sort of the branching extent that you normally might see in I'll say a large Pammersian, like we see in the Gulf of Mexico, or we've seen in the canyons, in the seamounts around here. These seem to be almost just small, I'm not going to say single stalks, but very few branches, right? Very few sort of lateral growth. The extent's not much, and it's noteworthy here. And I guess in the back of my mind, I wonder if it is the same species we've seen elsewhere, if it's something different. You never know,
Yeah,
right?
no,
Until
right.
you look at them closely.
Yep, could be something different. It's a very good point, and I can't stress that enough. We've gotten some really good imagery, and again, doing the best we can with about 30 scientists online in terms of the fauna that we see in the deep sea. But to get confirmation on a certain species name, it's always a must to have a collection.
Like another bamboo coming up.
Oh, yep, in the back. And then is that an Anthomastus on the left? Looks like it. And is that a bivalve? Dave, you see? Or is that an anemone right above your lasers? It's like blown out with the light. Yep. Yep, an anemone. And then frame the coral, please. Just two red shrimp. And then we'll want to go in and take a close zoom where it branches in three directions.
It also looks like another Gorgie here down left.
Oh, yep, I see that.
Looks like, I'm not sure.
Oh, with an I. Mm-hmm. It's a small Metallogorgia. Is that potentially another juvenile? Is there? I saw you mention that earlier, Scott. Is there different growth forms with juvenile Metallogorgia?
Yes, the Metallogorgia quotes adults would have that dandelion-like burst of branches at the very top of the colony with a long stalk about 1 meter tall with no branching on that stalk. Here you can see that there are branches arising along the stalk before you get to that top tuft. That's why we call them juveniles.
And do they lose those?
As they age and grow taller, they drop all the lower branches.
Okay, we're getting a close-up of Metallogorgia and a bamboo coral.
So you can see that top crown is not well formed yet, and that ovuroid's not up into that crown yet either.
Yeah, that's interesting observation. Right.
Yeah, that's a good point that Tim makes. The ones we were seeing earlier had a pretty well-developed crown and some lower branches, so you could think of more of those as in their teenage years where this is younger. The other difference is in the adult colonies, that ophiuroid, that brittle star would be in the crown and not lower down on the colony.
Right. So the expectation is that that opioid would then move up to the crown at some point. But well, perhaps when the other branches are shed.
Okay. We can pan out. And we're at a depth of 1722. Our temp is 3.77. And we're really here at a large debris the rubble scattered at the base of the wall. This is a field of large blocks. And this suggests that there's multiple phases of failure along this part of the canyon wall. Okay, and we are at waypoint 1. I think we're going to continue right on up to waypoint 2. How does that sound for everyone?
Sounds great.
Sounds good.
Oh, a large skate. Getting a really good image of a skate moving vertically up this wall face. And we're zooming in. And we're getting imagery of a ragged skate. Our depth is 1720. Local time is 12:58. Another cutthroat eel. Fly trap anemone. Can you zoom on that fish? Is that a cutthroat? That fish? Oh, right. It's a cutthroat. That's cool. Thanks. Yeah, that's good. Thank you. It looks like a grenadier macraride of some type. Paramerissia, another sponge. It's Anthomastus. Another bamboo coral at the top of this rock block. And can you snap zoom on the red and the fore of the front of this rock? Front of rock. Ooh. I just hit my elbow. And I thought I might have hit Roland, but he had gotten up. Another Bathypathes, and there is an associate. In the background, we have an anemone.
I think there might be a Parentopathes black coral just behind the Bathypathes. It's really
Oh, I
small,
see it. Yep.
slightly to the right.
And then a zoom on associate. Another
Yeah,
image of
Andrea,
a black coral. Oh, go ahead.
it appears to be an ophiuroid, doesn't it?
Yeah, I
This
think
is kind of
so.
rare to see on a Bathypathes, if that's what this is. We really don't see that. At least I haven't seen it.
Yeah, I haven't seen that either. And I want to note the other black coral in the back, the tiny one. I'm not sure if that is a Parentopathes or a very small Bathypathes. It's got one, it just looks like there's one polyp coming off the branch.
Yeah, I don't think that's a Parentopathes.
I agree. Very, very small Bathypathes.
Okay. All right, that's good. Is that a Paramerissia? Again, that single stalk. Oh, there's one branch off the side, but then another sponge and several holes in the rock here. Again, heavily bored. Yep. Okay, and then when we move up over this rock, Dave, get a snap zoom on the bamboo coral. Yeah, and Jason Chater is noting that there appears to be heavier bioerosion than below. Yeah, you can zoom in on that. And the sponge here. There's a few brittle stars. 1, 2, 3. Maybe a shrimp on the right. And we're getting image of a glass sponge, again, translucent. Highly lobed, a lot of structure. Go ahead. No, I think we can continue moving. I'd like to make it up to waypoint 2. And let's frame sponge and then frame coral. And is that a crinoid at the base? Of a very large sponge.
Andrew, another thing here to the right, you're just seeing how it looks like a large, well, what was that? What is this falling down? You get a hold fast there at the beginning.
Oh yeah. Sponge on the seafloor. And this bamboo coral has, different branching pattern, although maybe the same species, potentially. But the branching patterns do change from individual to individual. And then if we could zoom in on a split in the branch.
Looks like keratoisis, similar to what we saw before, but agreed the branching pattern is slightly different.
Okay, thanks, Dave. And several more Paramyrcia colonies on top here. Our depth is 1721 and local time is 1308.
Sure. Good bit of debris. Can you do a quick zoom on some of that debris, pilot, please? Oh yeah. So this is a huge debris field of large debris field of cup corals, all dead, all littering the bottom, which suggests that we probably have a huge or a good wall face in front of us. Yeah.
That's pretty incredible.
That's very large.
up here.
Nav, can you drop a target, please? DSC2, I think we're at. Thank you.
Jason, could you repeat?
Oh, when you get a chance, could we get a view from Sirius here?
Sure.
This is where
Thanks.
the living ones are.
Video, could we get a view
Seeing
of
some cup
Sirius on
coral on this
I2?
wall.
So this wall has several live cup corals and several dead below.
If we can get a zoom on some of the ones on the wall, please, pilot.
Yep. Zoom in.
Thanks.
Here we are. Not quite so much. Come back out.
Sponge.
There's a few live ones here.
Yeah. Mixture of live and dead on the wall.
Desmophyllum.
Yes.
Are those sponges, the covered in
Anemone.
the anemones? Can we get a zoom back where you were? Oh. Ooh, we cannot.
Yep. Nice overhang.
That's all right.
That's why they're all there.
Wow,
Yep.
that's amazing. Large overhang of live and dead cup corals and several scattered dead cup corals on the bottom. Coral rubble. Very nice feature, but not very good for the ROV to be under that.
It's all right. We're okay now. I just get myself some.
I'm all done with the Sirius view if you need to switch back.
I think we could keep it on for a little. It's a pretty good view.
It's a pretty amazing view.
Yeah.
Tilt down here. Sorry about all the dust.
Hey, Andrea, this is Tim.
Go for it, Tim.
A lot of time.
Yeah, we got some of these round, I'm going to say sort of olive green drab sponges here. We've seen quite a few of them.
Yep, with the white in the
Maybe
center.
those too.
Yep.
Yeah, I think so. There may be some sediment on them.
Yeah. Let's see if Dave will do that.
So science, just to preempt that a little bit, if we want to go back down there, we got to back up the ship. As you can see, as I back up, it's kind of pull a little
Do you hear that, Tim? We'll have to back up the ship if we want to go back down there.
basically stumbled into a little maze.
Sorry,
Nice.
Andrew, you talking to me?
Yes, we'd have to back up the ship if we want to go back down there. We kind of got caught up under that ledge and ship's moving. Do we want to do that to get an image of those sponges?
What, is there one right here on the edge? I think I was
Could you just do a zoom in
just below us.
into the ledge?
Yeah, very request I'll stop the ship.
Oh, we're stopping the ship.
Let me have a look around, Andy, and then we'll,
Cool.
we'll get back to it here.
Awesome. Thanks, Dave. Thanks, Brian.
If you look at this Sirius view, there's a bunch of mountain in front of us, so
Right. Pretty rugged terrain here.
And this is exactly where we'd expect it based upon the maps that we were looking at last night in terms of slope and stuff. So we're getting, yep.
so as soon as Sirius stops moving, I will happy to have a look around here.
Great, thanks. So we are,
So do we, I didn't touch it, do we want to go back down in there?
yes.
Okay. It'll take us a minute to set up.
Okay.
Have they stopped? So the ship is stopped. Okay.
Ship is stopped.
Sirius is not. Yeah, that's what I thought. Okay. So the view from for the co-pilot is still creeping in on me a little bit here. Unless you're down at like 67 degrees. Yeah, okay. Take it down just a little bit more. I can fool around the top edge here until about 75 degrees. That's pretty good right there. We'll work with this for a minute. Okay. Just holler at me if I cut you off in which direction. Okay.
These cup corals extend right up to the ledge that overhangs the circular area that we'd found them all in. Looks like there's a bamboo coral on top of that ledge.
That's correct. Thank you, Rich. Video exposure.
We're at a depth of 1709 and a bottom temp of 3.7. Local time is 13:15, and we're just viewing here cup corals, both live and dead. They're a type of stony coral, scleractinian coral. They're colonizing a small failure scar. Down below this area, there was a field of dead coral, cup coral, and we often call this a rubble. So, dead coral rubble on the seafloor beneath this heavily colonized scar here. And we have a couple sponges, a different sponge species, hexactinellid, and then we're really interested in those brown sponges, getting a closer view on those.
From here, I can zoom from this position, but I'm not going to disappear.
Perfect. That's totally understandable.
Looks to be about a meter from the flood. Yeah. Okay, a little bit of zoom, please, Joe. That's good right there. We'll work with that for a minute. A little more zoom.
Whenever you're ready, you
So
can
this
zoom
looks
in
like
further.
it might be a demo sponge. It could be a geodeid.
A geodia, demo sponge.
I'll enter it into the chat room log.
Okay. And so sponges are in the phylum Porifera.
There's cup corals right underneath it.
Good, Rich. Thank you, Rich.
And if we zoom out and move left, there's another coral or another sponge to get an image of.
There's an off, please, Joe. Oh, that right there. How far left are we going?
Not sure where. Did you see it?
No, I didn't see it.
There's one to the
So,
right below.
Can you zoom out?
Oh, wide.
yeah, go full wide.
There's this one over here to the
Yep,
right.
Yeah,
that
go.
one.
That one. Okay, back to the previous zoom, please. Hold that. Thanks. Down over.
Yeah.
Tony, I see your pilot camera, iris. Let's go to auto iris or auto iris. Trying to get a look at the top of the RV for you. Yeah, it doesn't look up horizontal as high as you can go with that. We'd have to use HE2 if we want to look at up at the overhang, which
All right, you can zoom in whenever you're ready.
that's macro video.
All right. Already at
Looks like it's made out of cheese cloth.